Hello, good afternoon, everyone. I'm Betty from RWD Life Science. Welcome to our webinar in RWD Academic. This is a platform for global researchers sharing research achievements. And today is our first live course of 2022. And we are honored to have Dr. Juan Pablo de Rivero Vaccari. And he is a research associate professor in the Department of Neurological Surgery, the Miami Project to Cure Paralysis in the University of Miami, Miller School of Medicine. He has studied the innate immune response for over 15 years. And he was the first one to show the involvement of the inflammasome in the steroid event. And today he will bring us an academic sharing about inflammasome regulation following traumatic brain injury. TBI affects approximately 10 million people worldwide and has become a major cause of death and disability. Inflammasomes as immune signaling complexes plays a key role in the TBI research. And from this webinar, we will know the mechanism of inflammasome in the innate immune response after TBI, and the reliable markers found from the TBI patients, and also the system effects of TBI. And hello, Dr. Pablo. Please say hi to everyone.
Hello, everyone.
Hello, Dr. Pablo. And please start to share your screen.
Will do. Can you see it now? Perfect. Yes. Hello again. Thank you for the invitation. Thank you for the opportunity. And today we'll talk about our work on the inflammasome and traumatic brain injury. First of all, I'll disclose my conflict of interests. I'm a co-founder of Inflammacore, and I'm also a scientific advisory board member of Diverse Therapeutics. So what is traumatic brain injury? Traumatic brain injury is a result of an external force to the head that results in a linear acceleration of the brain or a rotational acceleration of the brain, as well as could be an impact deceleration. In the United States, most traumatic brain injuries are mild in its severity, accounting for 80% of the cases, whereas 10% correspond to mild and 10% correspond to moderate. Traumatic brain injury can be classified in different ways. Here we use, it could be mild, moderate, and severe, as I mentioned, but we could use the Glasgow Coma Scale to differentiate these three different categories, which it's the GCS, or Glasgow Coma Scale, is assessed within the first 24 hours after TBI. You could also classify it based on the loss of consciousness, as well as alterations in consciousness, whether it is up to 24 hours, more than 24 hours. We could also use imaging modalities to determine whether the brain looks normal or abnormal. And also the length of post-traumatic amnesia could be also used to classify traumatic brain injury, with mild cases ranging, the PTA ranging in mild cases from 0 to 1 day, and for more than 1 to 7 days for moderate, and beyond 7 days of amnesia for the severe cases. So that's how we assess the injury severity. Now, we also assess mostly this injury severity clinically with the GCS, or Glasgow Coma Scale, as I mentioned, and it has 6 different categories, with 1 being the worst outcome, and then the 6 being the most severe type of severity of TBI. Which means basically, for instance, a patient that presents a GCS of 2 eye openings, they could open the eyes to pain, their speech is incomprehensible, and they have some motor deficits. Now, in terms of determining the outcome, which normally we do it at 5 or 6 months after traumatic brain injury for our research studies, we use the 2 different scales, the GOS or the GOS extended. The GOS goes 1 through 5, with 5 being the best outcome, and the GOS extended, we do it goes from 1 to 8, depending on the patient's response. And normally when we look at when we think of traumatic brain injury, we think of it as an acute type of insult. However, there are long-term consequences to traumatic brain injury. For instance, there are neuropsychiatric complications associated with TBI, such as major depressive disorder, anxiety, PTSD, psychosis, paranoia, pseudovulvar affects, as well as aggression. So there's this variety of symptoms and signs that will arise after traumatic brain injury. That's why some patients with trauma tend to have different a change in personality after TBI. Now, if we look at the anatomical characteristics of TBI, just going a little bit deeper, the actual trauma to the head and the impact, whether it's a deceleration or rotational change in the mass of the brain, results in the level of the neurons. Here, you can see that there is a disruption of in the microtubules, which is associated with also the leakage in contents through the membrane that blocks exoplasmic transport of trophins, and also there's a result of increase in cytokines that we'll talk later about. But then there's a disconnection in the axons, which impairs synaptic function, and there's also accumulation of axon injuries, such as accumulation of amyloid protein, amyloid beta, presenilins, and this leads to an effect on lowering the an imbalance in energy, and there is also activation of calpains, caspases, as a result of calcium imbalance, which leads to cytoskeletal proteolysis, and this eventually results in the loss of function that I just described before. But when we look at the cellular mechanisms, going a bit deeper, see that disruption to the membrane causes axonal damage that affects neurons to affect the neighboring cells, as well as neighboring cells start to affect neurons as well. So for instance, we have a variety of cells playing a role here. You have astrocytes, we have microglia, we have endothelial cells, and that all are contributing to the molecular mechanisms of the pathophysiology of traumatic brain injury. For instance, astrocytes, which are swollen, and then they manifest, that swelling manifests as edema, are associated with production of glutamate that goes on to activate glutamate receptors and the receptors in neurons, causing excitotoxicity. Then neurons start to die, and they release these pattern recognition receptors, which in the case of TBI, they are referred to as danger-associated molecular patterns, or DAMPs. And they start being secreting cytokines and chemokines and then neutrophils as well. So this whole environment orchestrates to increase oxidative stress, which also goes on to activate cell death, such as necrosis, apoptosis, pyroptosis, which I'll mention later, and all that is part of the inflammatory immune response. So when we look at the stages of TBI from minutes to years, we see that there are several events that take place. We see that there's neuronal injury, blood-brain barrier damage, infiltration of and activation of myeloid cells, neurodegeneration that leads to chronic traumatic encephalopathy, cell death, as I had already mentioned, such as apoptosis and demyelination, autoimmunity. Then there's also DAMPs, cytokines, and chemokines that are secreted to activate these pattern recognition receptors. Neutrophils get activated, monocytes get activated into macrophages in the injury site, and then there's also obviously activation of microglia, astrocytes, B cells, and T cells. And you can see that most of it happens within minutes to 2 days, and then for weeks it starts to decrease. But then there is this autoimmune process also mediated by T cells that is also present in the weeks to years after TBI. So going into inflammation, the inflammatory response is divided into 2 phases: the innate immune response and the adaptive immune response. I personally studied the innate immune response, which is characterized by pattern recognition receptors like toll-like receptors, node-like receptors, and rig-like receptors that activate these complexes to secrete cytokines, within minutes to hours after an insult, and then they go on to activate T cells and B cells and mount an adaptive immune response. So these node-like receptors form cytoplasmic complexes, such as the inflammasome. And we have also other pattern recognition receptors, like toll-like receptors, that are present in the cell membrane. Particularly, the inflammasome is a multiprotein complex comprised of caspase-1 and caspase-5 in humans, NLRP1 or NAP1, also NLRP3, which these are the two most understood inflammasomes, and the adapter protein ASC. So these proteins come together to form an inflammasome complex that is responsible for the activation of the cysteine aspartate caspase-1. So when here is a depiction of the NAP3 inflammasome in the center, and which goes on to activate caspase-1. When caspase-1 is activated, pro-interleukin-1 beta and pro-interleukin-18 are cleaved into its active fragments, and then they are secreted through different mechanisms, one of which involves the cell-mediated, inflammasome-mediated cell death mechanism of pyroptosis. So the substrate of pyroptosis, or inflammasome-mediated cell death, is gasdermin D. And gasdermin D gets cleaved at its amino terminus, and the amino terminus gets incorporated into the cell membrane to form a pore through which cell contents, including the cytokines I just mentioned, get released. Also, this process is referred to as a canonical inflammasome, but there is also non-canonical inflammasomes that are activated, that are mediated by caspase-4 and 11 in rodents, caspase-5 in humans, and also caspase-8 has been involved, shown to play a role in inflammasome activation. Things that are involved in inflammasome activation are mitochondrial dysfunction, increase in reactive oxygen species, lysosomal rupture, as well as high extracellular potassium or potassium influx, and as well as the presence of high levels of ATP that bind to P2X7 in a pannexin-1 mediated process. So when we study traumatic brain injury, we use different models, and this is one of the models that we use, which is the precise impactor from RWD. And basically, for our controlled cortical impact injury model of TBI, we use this device where we place the animal in the platform, and there is a craniotomy that is made in the animal, and then there is a displacement that is controlled by this instrument. Other models that we also use in the lab are the fluid percussion brain injury and the penetrating ballistic TBI injury that mimics a gunshot wound. So for when we initially described inflammasome in the brain after injury, we used a model where we look at sham animals and animals that were sacrificed at different time points after brain injury, and then we obtained protein lysates to look at to carry a Western blot analysis. And here are the results of the immunoblotting, where we have animals that were naive, meaning that they did not undergo TBI and they did not undergo a craniotomy. Then sham animals that underwent craniotomy but not TBI, and animals that were sacrificed at different time points, 15, 30, 1 hour, 3 hours, 6 hours, and 24 hours after TBI. And then we blotted these membranes for of this cortical, containing this protein cortical lysates from these animals for caspase-1 and ASC. And as you can see, after TBI, you see an acute increase of inflammasome proteins in the cortex of rats in this case. So then, to determine, knowing that the inflammasome was activated, or at least caspase-1 was activated after TBI, the next thing that we did was to determine if indeed there was a multiprotein complex formed in vivo in the brain after TBI. So we immunoprecipitated with antibodies against ASC and NLRP1, and blotted for ASC, NLRP1, caspase-1, caspase-11, the inhibitor of apoptosis protein XIAP, and pannexin-1, and found an association between these inflammasome proteins, indicating that indeed in vivo there is there was an inflammasome complex forming after TBI. So then we used a therapeutic approach using a biologic where we had two groups. After injury, we treated the rats with either IgG isotype control or anti-ASC intracellular ventricularly, and then sacrificed the animals at 24 hours for Western blot and at 3 days for histological assessment. And it turns out that when we looked at the sham animals, the IgG treated animals, and the anti-ASC treated animals, that there was a decrease in the levels of caspase-1, which is quantified right here, consistent with lower levels of I1 beta and XIAP. And consistent with this decrease in inflammasome activation after treatment with anti-ASC, there was also an improvement in the mean lesion volume in the cortex of the anti-ASC treated animals when compared to the IgG group. Now, we looked at different cell types to see in which cells these inflammasome proteins are present. And we look here we show you neurons that, this is a confocal image of frozen sections that were double stained with the neuronal marker NUN, shown in green. And the inflammasome proteins, caspase-1, 11, ASC, and NLRP1 in red. These animals were sham animals and animals that were sacrificed 4 hours after traumatic brain injury. You can see increasing immunoreactivity after 4 hours in the inflammasome, in the TBI animals. And here you can see the increasing reactivity in caspase-1 and also this ASC protein expression in the cell cytoplasm of these neurons as well, similarly to NLRP1, indicating that inflammasome is present in neurons. Back then, when we started doing this work, we really didn't appreciate much whether the role of inflammasome in neurons, but now we know that also that obviously inflammasomes are present in neurons, microglia, astrocytes, oligodendrocytes, and endothelial cells, among other cells in the CNS. Now, therapeutic hypothermia is a therapy that is used in some places to treat patients with traumatic brain injury. So we went on to see if therapeutic hypothermia improves if part of the beneficial effects of therapeutic hypothermia after TBI is through inflammasome inhibition. So we have the animals that were not injured and animals that were normothermic and hypothermic for 4 hours after TBI, as well as 24 hours after TBI. And when we look at caspase-1, and then we plotted for caspase-1, 11, and ASC, and we saw that at 24 hours, compared to the normothermia versus hypothermia group, there was an increase a decrease in the hypothermia group for caspase-1 and caspase-11, consistent also with lower levels of interleukin-1 beta, cliff interleukin-1 beta at this time point due to therapeutic hypothermia, indicating that part of the beneficial effects of hypothermia are the result of inflammasome inhibition. And here we show a focal image of process sections also for the normothermia and hypothermia for NUN cells that were also double labeled with caspase-1 in red. And indeed, you see a decrease in immunoreactivity in the hypothermia treated group compared to the normothermia animals. And then here we used the penetrating ballistic brain injury model, which, as I had mentioned, it mimics a gunshot wound to the head. And then we looked at microglia and ASC in different regions. Here we looked at the contralateral side to injury, and two sections within the ipsilateral side, the perilesional area, as well as the intact dorsal area. And when we look at here you have the merged, the DAPI positive channel, the microglia positive channel, which was labeled with IVA1, and then ASC. And you can see that in the perilesional area, and to some extent also in the intact dorsal area, you see ASC present in microglia after traumatic brain injury. So when we look at microglia at different stages, from resting, prime, activated, and amyloid, the last two being the most active, involved in cytokine release and also in phagocytosis, as microglia, let me highlight, they are the professional immune cells of the CNS. So we looked at, obviously, that we compared contralateral versus ipsilateral, and as expected, IVA1 positive cells are present at higher levels, or microglia are present at higher levels in the ipsilateral side compared to the contralateral side. But when you look at the ipsilateral versus contralateral, also for ASC positive microglia, you see that there is a significant increase of this ASC positive microglia in the ipsilateral side. And when we look at the different stages, resting, prime, activated, and amyloid, we'll see that indeed there is more microglia, obviously, in the activated and amyloid in the ipsilateral side after TBI compared to the contralateral. But also, those cells were also immunoreactive for ASC, indicating that the important role that ASC plays after traumatic brain injury in these professional immune cells of the CNS. And then in other studies in humans, what we did for our biomarker studies was we collected cerebrospinal fluid and serum from patients after traumatic brain injury at different time points. And then we assessed the potential for these proteins as biomarkers of injury severity and injury outcome. So here is the results for the outcome of after traumatic brain injury. So we dichotomized the GOS extended or the Glasgow outcome scale into favorable and unfavorable outcomes. And it turns out that patients within the first 24 hours, after traumatic brain injury, ASC, caspase-1 and NAL1 or NLRP1 were elevated or were at higher levels in those patients that went on to have poor outcomes when we assess those outcomes 5 months after trauma. So acute elevations of inflammasome proteins in the CSF, and later we've also shown it in serum, even though they're acute, they have long-term lasting consequences for outcomes after traumatic brain injury, outcomes that we actually measured as far as 5 months. And then when we looked at different variables in features in another studies looking at caspase-1 in the CSF, we noticed that patients that have high intracranial pressure tend to have higher levels of caspase-1 at different time points, as much from day 1 to day 5. And similarly, when we measure caspase-1 levels in the CSF and did that again, favorable versus unfavorable, we also noticed that those levels were higher for those that had unfavorable outcomes, consistent with our previous results from previous studies. So it seems that caspase-1 plays also a role in intracranial pressure, particularly, or that intracranial pressure plays a role also in caspase-1 levels. So then we collected for different time points serum as well as CSF on patients after TBI. So we collected up to 5 days for and 3 collections a day. And we measured the levels of ASC and caspase-1 and other inflammasome proteins. And here we show you for these 2 proteins and compare them to control non-injured TBI patients. So you can see that acutely up to the 6th collection corresponds to the first 2 days after TBI. And then you see that there is an acute increase in ASC that starts to decrease on the 2nd day after trauma. And the same thing happens for caspase-1. However, it still remains elevated compared to control levels. With this, we used different collection points to assess biomarker potential of inflammasome proteins. And in this case, we looked at, when we look at a biomarker, we assess different things. We calculate the receiver operating characteristics, which is a plot of the sensitivity versus 1 minus the specificity. And with that, we obtain an area under the curve. And the ideal biomarker has an area under the curve of 1. So biomarkers above 0.7 are considered good biomarkers, but above 9, above 8, those are even better biomarkers. Ideally, above 9, it's a very nice biomarker with nice significance, sensitivity, and high specificity. And consistent with this concept, we found that already in the 2nd collection, we were able to see that that caspase-1 as well as ASC in serum had high levels, high area under the curve values, consistent with the good biomarker potential that that inflammasome proteins have after traumatic brain injury. And then we did the same thing on CSF, but in the case of the CSF, what we found was that ASC and interleukin-18 were those cytokines that were highly elevated after traumatic brain injury compared to the control group. And again, you can see that obviously we get closer to the brain, so the area under the curve for ASC particularly was much higher in the CSF than for serum. However, being able to identify fluid biomarkers like serum, urine, tears, CSF, if you can have enough sensitivity to look at a biomarker in serum, then you don't have to go into very invasive assays or tests like CSF collection, which is done through a lumbar puncture. So that's for that reason we always try to look for serum biomarkers that we can actually monitor disease, injury severity or from injury prognosis, as well as response to treatment. And then here we show you images of exosomes. We isolated exosomes from control subjects and spinal cord injury subjects on the left side and bottom, and then also traumatic brain injury subjects. And compared to controls and also to patients that had poor outcome, as here as this patient, patient 2, and patients with good outcome, patient 1, and then patients 4 and 5 here also with good outcome, and patient 3 with a poor outcome. When we compare this for spinal cord injury, which was measured which was assessed with the Asia score at discharge, and then the TBI was assessed with the Glasgow outcome scale, what we found was that in those patients that did poorly after a spinal cord injury and traumatic brain injury, that the inflammasome proteins were enriched in exosomes or extracellular vesicles. So that was interesting, showing that that you have these vesicles that are released after traumatic brain injury, and that they contain inflammatory proteins, particularly inflammasome in this case. And this led us to start looking at the systemic effects of traumatic brain injury. So what happens is that after traumatic brain injury, yes, definitely, obviously, there is effect in the brain and the CNS, but there is also effects on the thyroid with the levels of, let's say, triiodothyronine. There is also effects in the heart, which increase oxidative stress, increase troponin, decrease left ventricular ejection fraction, decrease heart rate. There's gastric reflux, delay emptying of the stomach. There is also increased glomerular filtration rate, decreased clearance, so overall decreased renal function. There is also an impairment in or lower levels of luteinizing hormone, follicle-stimulating hormone. At the level of the testes, there is a decrease in testosterone. There is also increased permeability of the gut and intestines. There is also increased edema, inflammation, and in the liver, there is an increase in hepatic enzymes, also increased hepatic metabolism, and in the lungs, which we also started to look at the inflammasome in this role, there is also acute lung injury. So many of these patients after traumatic brain injury, some of them, especially after severe TBI, they have acute lung injury, and they also have a tendency to look at have cardiac abnormalities. So we hypothesized that exosomes are released from the brain containing inflammasome proteins, and that they reach target organs or cells that then they go on to activate the inflammasome in those cells, and then that results in inflammation. So we looked at, for instance, in the context of the target cell being the cells in the lungs, we looked at ASC in the serum of and extracellular vesicles of patients with TBI and control subjects, and we've had already anticipated it based on our previous studies. ASC was elevated in the serum as well as extracellular vesicles compared to controls. But when we divided those patients that had acute lung injury and TBI and no acute lung injury and TBI, we noticed that those patients that had acute lung injury and TBI had lower levels of ASC in the serum as well as the extracellular vesicles, suggesting a role for these exosomes in the pathology of acute lung injury after TBI. And here we show you the lungs of TBI mice that were injured. Well, here's sham and TBI mice that were injured at 4 and 24 hours. They were injured and sacrificed at 4 and 24 hours, and then blooded for the inflammasome signaling proteins caspase-1, ASC, interleukin-18, AIM2, interleukin-1 beta, and the danger-associated molecular pattern, HMGB1. And as you can see, compared to sham, these proteins were elevated in the at 4 and 24 hours in the lungs of these mice compared to sham and injured mice. And here you see the confocal images of inflammasome proteins in the lung tissue. Here you see the alveoli having inflammasome protein expression at higher inflammasome protein expression in the TBI group at 24 and 4 hours compared for the proteins of interest in AIM2, caspase-1, and ASC when we compare them to sham and injured animals, indicating that in the lung of these mice after TBI, there is a role for inflammasome in contributing to the inflammatory response that potentially could lead to acute lung injury. So then we used an inhibitor of the inflammasome known as that we're developing called IC100, which is a humanized version of anti-ASC that we used in the past. So basically, this is an anti-ASC antibody, and then we treat animals with IC100, which here corresponds to anti-ASC, and then another group of animals were treated with enoxaparin, which is a blood thinner, similar to it's a low molecular weight heparin. Then we have untreated animals, we have sham saline animals, and then naive animals, which were blooded for caspase-1. And then the lung tissues were blooded for caspase-1, ASC, interleukin beta, A2, and HMGB1 again. And as you can see, the untreated animals had higher levels of this inflammasome signaling proteins compared to the enoxaparin and the anti-ASC treated mice. When we did the histology, here you can see the sham, you can see the alveolar space, which is normal, like it's shown here in the saline animals, in the saline sham animals. But then in the untreated, you see the decrease in the alveolar space, which was improved in the enoxaparin and the anti-ASC treated group. And here we quantified this histology based on an acute lung injury score. And according to the acute lung injury score, the untreated animals had much higher levels of, had a poorer histopathological outcome when compared to the enoxaparin and the anti-ASC treated mice. So then, just to see if indeed extracellular vesicles can induce inflammation in lung cells, we isolated extracellular vesicles or exosomes from TBI patients, and then we treated human pulmonary endothelial cells in culture with those exosomes, and as well as another group of cells were treated with exosomes from control subjects. And then we blooded those cells for caspase-1, ASC2, and HMGB1. And the systematic analysis indicated that indeed, with the group of cells that received exosomes from TBI subjects had lower levels of caspase-1, ASC, into HMGB1, and interleukin-1 beta, indicating the role of these exosomes containing inflammasome proteins in spreading the inflammatory response that results from TBI to other organs in the periphery, such as the lungs in this case, which is responsible for acute lung injury in severe TBI. So what I've covered today is that the inflammasome is activated in cortical neurons after traumatic brain injury. We can use anti-ASC, which these days we're developing under the name of IC100, to inhibit inflammasome activation in the brain of rats after TBI, which results in improvement in histopathological and functional outcomes. Part of the beneficial effects of therapeutic hypothermia after traumatic brain injury could be associated with its role on inflammasome inhibition in the cortex. And we've also today I've also shown that to you that ASC is elevated in microglia after TBI, indicating that this cell type is an important contributor to the inflammatory response through the inflammasome to the pathogenesis of traumatic brain injury. Also, that these inflammasome proteins present in the CSF and serum, they have a high potential to be good and diagnostic biomarkers of injury severity, as well as prognostic biomarkers of traumatic brain injury outcomes. Just by looking at acute changes of inflammasome proteins, it can actually be a good prognostic indicator of what may happen to patients with just with today's technology. To look at patients 5 months after trauma, based on the Hive area underscore values, which are desirable for these biomarkers in the use of personalized medicine. And then finally, we've shown that extracellular vesicles in TBI patients contain these or are enriched in inflammasome proteins compared to EVs from healthy individuals. And the DCVs containing inflammasome proteins are capable of causing inflammation in peripheral organs, such as the lungs, which in the case of the lungs causes acute lung injury. And we've shown that mechanistically by also showing that inflammasome is activated by extracellular vesicle delivery in human pulmonary endothelial cells. I want to finish by acknowledging my laboratory members, Ben Asir and Roy Haddad, as well as my collaborating laboratories, Dr. King's laboratory, who I did my PhD with, and then my collaborator in Spain for John Perez Bárcena for our work on biomarkers, as well as Dr. Dietrich and Dr. Bramlett here at the University of Miami. With that, I want to thank you all for paying attention.
Thanks very much. Thanks, Dr. Pablo, for your very detailed sharing. And so goodbye, Dr. Pablo.
My pleasure. Thank you for the opportunity. Have a great one.
Okay. RWD products like process impactors, cell testing instruments, laser sparkle imaging system, and system machine are in hot trials now. Welcome everyone to search our website and have applied. And once again, thanks Dr. Pablo's wonderful course, and see you next course.